So this is the third in the series on the kidney today, and just as you can see, an overview of the information that we have covered over the past two. So if we just zoom in quickly, see the first one looked at the growth structure, and you see the cross-section through the kidney, which we discussed in detail, and also the functional unit of the kidney, the nephron. Now last time, as you can see highlighted there, we looked at that three-part filter through ultrafiltration, and in terms of that we talked about the three-part filter. So if there is anything there that you're thinking, I can't quite remember the growth structure, or I'm struggling on that three-part filter, you need to go back and have a look at that. For today's session, we are going to look at the selective reabsorption, which will be the third in this series that we look at. So in relation to selective reabsorption, we need to now focus our attention on the proximal convoluted tubule, which you can see here highlighted with this red box that I have put around the proximal convoluted tubule. So this is part of that functional unit of the kidney, the nephron, and it is the first part of the nephron directly after your Bowman's capsule. It is the site of selective reabsorption, and in relation to this, your kidney is going to be filtering up to 180 litres of liquid per day approximately, and 1 to 2 litres of that is then leaves the body as urine. So 178 litres has to then be selectively reabsorbed, and the first stage of that is taking place at this proximal convoluted tubule. So we need to have a look at the cells that exist here at the proximal convoluted tubule, and we need to look at exactly what is happening. This is the site at which we're going to, by the end of this tubule, have reabsorbed all of our glucose and all of our amino acids and a small amount of your H2O, your water. So if we start to look at the proximal convoluted tubule, you can see here I've just drawn very roughly your proximal convoluted tubule cells, which are going to line this tubule. We can see the filtrate from the Bowman's capsule is going to sort of travel down through this tube. It's convoluted, meaning that it is wiggly effectively, so it's not a straight line, it is a wiggly tube that we have that we're looking at. So we need to look at the adaptations to start with. The first adaptation to note are the microvilli. So you can see here, I've demonstrated that with that brush border down those proximal convoluted tubule cells, and this is there to increase the surface area so that you've got a large surface area for reabsorption to take place over. The second adaptation is that there are lots of proteins embedded into the membranes of these cells, and these comprise of both sodium-potassium pumps that are going to be constantly actively transporting and sort of maintaining concentration gradients to enable facilitated diffusion to take place, and there are also then co-transporters to enable this facilitated diffusion. As we have lots of membrane proteins, we also need to have lots of ribosomes in order to make those proteins, and we also need to have lots of mitochondria in order that we can produce ATP to enable the active transport to take place. So the next thing that we need to look at then is how we use facilitated diffusion at the proximal convoluted tubule in order to ensure that all of the glucose and all of the amino acids are completely reabsorbed before they reach the loop of Henle. So as you will see, I've just drawn for you, you can see the cells of the proximal convoluted tubule and the filtrate that would be moving down the middle, and then adjacent to that on the left you can see the capillary. We're just going to look at how we can reabsorb those amino acids and your glucose here. Now, to start with, we have to go back and think about facilitated diffusion here. And we're going to start with the sodium-potassium pump, which is on the left-hand side. And we're going to move the 3 sodium out and then 2 potassium in. So once inside the cell, the amino acids and the glucose can then diffuse across the proximal convoluted tubule cell and then out and into the capillary, and we'll just show a protein channel to display that on the screen in a second. So one thing just to note here is that the fact that you have had the sodium moving and the change in that concentration is going to have an osmotic pull. So you are also going to see the movement of water, and that is going to move into that cell just following that osmosis. So as you see the sodium being brought back in using facilitated diffusion, the water will then move by osmosis into that cell as a result of that. So just to recap very quickly then, we're at the proximal convoluted tubule today looking at selective reabsorption. Now these cells are adapted for reabsorption because they have got that all of those microvilli which create the brush border that increases that surface area. There are lots and lots of proteins specifically for moving sodium and potassium, also co-transporters for facilitated diffusion, and then some protein channels to enable the movement of the glucose and amino acids once they have moved into the cell. We see the filtrate moving down at this point. That filtrate has got everything that was small enough to fit through the three-part filter. And at this point we need to reabsorb all of the amino acids and all of the glucose. We do that by setting up a electrochemical gradient using our sodium-potassium pumps. So 3 sodium out and 2 potassium in. We then use that electrochemical gradient of the sodium using a co-transporter to bring in either the amino acid or the glucose. Once that has gone into that cell, that can then move out via a channel protein into the capillary and has then been reabsorbed into the blood, reminding ourselves that because of that movement of the water, of the sodium across the cell, that will then enable water to move via osmosis as a result of that also into the cells of the proximal convoluted tubule and then into the capillary. So there will be some uptake of water or reabsorption of water at this point, but the majority of that is going to be further on in the kidney, as we'll detail in the next video.